So, hello, let's continue where we left off in the last episode. In the last episode, we studied about a basic overview of a cell, right? Just a basic overview of cell. We studied what a cell is, basic definition. Then we studied about cell theory, like a theory that is true for all living cells in the world. And then we studied about a basic overview of any cell, right? Now we are going to classify cells. We're going to classify cells on the basis of whether they have a membrane bound nucleus or no. If they don't have a membrane bound nucleus, if cells don't have a membrane bound nucleus, then those cells are called prokaryotic cells. If cells do have a membrane bound nucleus, they are called a eukaryotic cell. Now, like we discussed in the last episode, eukaryotic, eu means true and karyon means nucleus, so it's true nucleus, that means a membrane bound nucleus, and prokaryotic means pro means primitive and karyon means nucleus, so it is a primitive nucleus. That is why it is having no nuclear membrane around its genetic material. Now let's go in depth in understanding what prokaryotic cells are. Prokaryotic cells, essentially, you might have all heard about bacteria, right? Bacteria. So prokaryotic cells are all bacteria. All prokaryotic cells are. Now this line is essentially not written anywhere explicitly in the NCERT, but it has always been hinted. It has always been hinted in the text, and I think it is very true. I think it is true. And as far as I researched, I read the whole text, like the whole book, whole 2 books of biology, 11th and 12th. Also, I searched it up on Google, and I think all sources point to the fact that all prokaryotes are bacteria, and all bacteria are prokaryotes. Also, one more important thing, all prokaryotes are part of kingdom Monera, which is a single kingdom. So all prokaryotes are part of only a single kingdom, and all organisms in kingdom Monera are prokaryotes. So this is a very important line, although not written anywhere explicitly. See, whenever I'm telling you something that is not written in the textbook, I'll be going in depth. I'll be trying to search it from every source possible, and then only coming and putting it up to you. So this is very, I think this is very true. This is a very true statement. So if you want to remember it, you can remember it, or else you can just forget about whatever I said. Okay. Now, prokaryotic cells. Let's read the text. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. Now, blue-green algae is also called cyanobacteria. Hence, blue-green algae is also a bacteria. It's a very primitive. It's one of the earliest living forms, life forms on Earth. Blue-green algae is very, very ancient, I must say. And blue-green algae, bacteria, mycoplasma, and PPLO. Mycoplasma and PPLO are also bacteria. Now, they are generally smaller. Prokaryotic cells are generally smaller and multiply more rapidly than eukaryotic cells. Now, there is a reason why they multiply more rapidly. First off, it doesn't have any compartmentalization as in the case of eukaryotic cells. Eukaryotic cells have different compartments, and for them to divide, they must replicate that exact compartment in both the daughter cells. So that takes much more time. Whereas in prokaryotic cells, they don't have such compartmentalization, so they divide more rapidly than eukaryotic cells. Okay, they may vary greatly in shape and size. The 4 basic shapes of bacteria are Bacillus, Coccus, Vibrio, and Spirillum. Okay, that's the basic shapes of now. Notice how they are saying the 4 basic shapes of bacteria are. Now, that is because prokaryotic cells and bacteria are essentially synonymous. They're the same things. All prokaryotes are bacteria, and all bacteria are prokaryotes. Hence, they can just say shapes of bacteria, but this is actually a topic, a line under the topic of prokaryotic cells. Now, The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. Now, while it is fundamentally similar because all of them don't have a membrane-bound nucleus, and and all of them don't have any membrane-bound organelles. So they are all fundamentally similar, but there are different types of prokaryotic cells performing different functions, and hence they have different shapes too, but they're fundamentally similar, and this too is a reason why they multiply more rapidly, because they're fundamentally similar and simple. Okay, now all prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. As we have already discussed, mycoplasma does not have a cell wall surrounding its cell membrane. Hence it can change its shape, because cell wall is a rigid structure. It gives shape to the cell, and if a cell does not have cell wall around it, the shape of the cell can change. So hence we call mycoplasma pleomorphic. So we call mycoplasma pleomorphic because it does not have a cell wall, and hence it can change its shape. Okay, so here we are reading that prokaryotes have a cell wall surrounding the cell membrane. Okay, so cell wall. So let's just draw along like however they describe the prokaryotic cells. Let's just draw along how they are showing the prokaryotic cell. Let's say this is the cell, and they're saying that it has a cell wall surrounding the cell membrane. Now cell membrane here just implies plasma membrane, as Sir Theodore Schwann had pointed out in his initial discoveries that animal cells have a delimiting structure as plasma membrane. So here this is a bacteria has a cell wall surrounding the cell membrane is plasma membrane. So let's say this is the cell membrane, this is the cell membrane, let's say this is the cell membrane, then this would be the cell wall. Now we learn further that there is an additional wall around all this, but that we learn in the coming subtopic. Okay, so this is the plasma membrane, this is the cell wall. I'm not drawing the whole cell wall, I'm just trying to express that it is a cell wall. So this is a cell wall, and there is another, it's a three layered cell envelope in bacteria, but in mycoplasma there is no cell wall. Okay, that is important, that's why it's pleomorphic. Okay, the fluid matrix filling the cell is the cytoplasm. Okay, that is true for eukaryotic cells as well. Now there is no well defined nucleus. Okay, no membrane bound nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA, and these smaller DNA are called plasmids. Now here there are so many new terms that have been introduced. First is genomic DNA. Maybe you don't know what genomic DNA is. And then there is plasmid, and then there is a non-genomic DNA, which is plasmid. Plasmid is a non-chromosomal DNA, right? Okay, now I'll try to explain, but we are going to go in depth into understanding all these genetic material stuff in cell cycle and cell division, which is a very beautiful chapter, which is like the next chapter, like the third chapter from now. But for now, let's understand what genomic DNA means. Now this bacteria dividing into two bacterial daughter cells, this parent bacteria, let's say it's dividing into two bacterial daughter cells. So the information required to replicate the parental characters into the daughter cell, that information is present in the genomic DNA. Okay, that information is present in the genomic DNA. So essentially genomic DNA is the main DNA of a cell. Main DNA, you can just understand, it's the main DNA of the cell, which is passed down from parent to progeny, and which is very important in forming the structure of the daughter cells from the parent cell. Also genomic before cell division, I may be going in depth into this, but I hope you can understand, so that because when we interlink and study, it's very beautiful. We don't have to remember much things, we just understand. So genomic DNA, before cell division, the genetic material of a cell is just present like randomly. It's just present randomly. It's just present randomly. Okay, in the cell it is just present randomly before cell division. But just when cell division starts, this randomness disappears, and these randomly arranged genetic material, it condenses to form chromosomes. Yeah. Why do they form chromosomes? So that genetic material can be equally distributed into the daughter cells. Okay, that is the the main function of why. Because see, if it is random, how can the cell equally divide this random genetic material, ensuring that both the daughter cells will have the exact identical information and all the necessary information that is needed to make the cell? That is not possible because it is random. Now, even if I divide it half, even if I just divide it half, one side will have lesser genetic material, the other will have more genetic material. And let's say even if they have equal genetic material, one side will have essential what do you say, essential genetic information, and the other side will have non-essential genetic information. So it's not possible for the cell to equally divide the genetic material if it is just present in the entangled form. That is why this entangled form disentangles and then condenses into chromosomes just when the in the very first step of cell division, which is prophase, right? So whenever prophase even start, I mean that prophase is the first stage of cell division. So yeah, the moment prophase starts, this disentanglement disappears. This entanglement disappears. It starts to disentangle and then condense into chromosome so that it can be equally divided into two of the daughter cells. Now, only genomic DNA, the point I'm trying to say is that only genomic DNA condenses to form chromosomes. Non-genomic DNA, which is the non-genomic DNA, can also be called non-chromosomal DNA because they don't form chromosome. Means if it is non-genomic, they will not form chromosomes. So example, plasmids are non-genomic DNA. If they're non-genomic DNA, they won't form chromosomes when the cell is dividing, whereas genomic DNA will form chromosome when the cell is dividing. So that is the difference between that. I'm just trying to explain the difference between genomic DNA and the non-chromosomal DNA or the non-genomic DNA. Okay, so plasmids are essentially very important in we'll go in depth into understanding plasmids in the biotechnology chapter of class 12th. It's very beautiful. Now, the plasmid DNA confers certain unique phenotypic characters to such bacteria, and okay, phenotypic characters to such bacteria. Now you might not even understand what phenotype and genotype is. Don't worry about it, we are going to study about it in the class 12th chapter of principles of inheritance and variation. It's a very good chapter, but for now you must understand I took the responsibility of explaining each and every line. So phenotypic characters, let's understand what phenotype is. Okay. Okay, so this genomic material, right, this is the genetic material. This genetic material, let's just zoom the genetic material and let's say this is the genetic material. Let's say, I'm just saying, let's say this is a human, this is for a human. Let's say this genomic material is for a human, and I took this genomic material from a human cell, and then just magnified the chromatin material, not the chromatin material, let's say I magnified the chromosome. Okay, let's say I magnified the chromosome, doesn't really matter though. So let's say I magnified it. Now what is a gene? You have to understand what a gene is, right? So gene is a segment of DNA which codes for a particular character in an organism. Okay, if this particular segment of DNA is coding for something in the organism, anything, it can be anything, it can be an enzyme, it can be for your any type of cell, like formation of any type of cell, it can be just anything, but it should be something. If this segment of DNA is coding for something in the living organism, then that segment of DNA is called a gene. Okay, a gene can have like different lengths, like I just drew two. That doesn't mean that exactly it is two. It is, I'm just doing it randomly to express what I'm trying to say, but it can be like of like this much and how much ever, but that segment of DNA must be expressing, must be contributing to the to the living organism. Means it should be coding for something that is found in the organism. Now this segment of DNA is called a gene. Okay, such a segment is called a gene, which codes for something in the living organism. That segment is called gene. Now let's say this particular gene is coding for my eye color. Let's just say that this particular gene is coding for my eye color. This gene codes for my eye color. Means this is the gene that decides my eye color. Okay, codes for eye color. Okay, so this is the genotype of the organism. The genotype for eye color. This segment is the genotype for my eye color. Genotype for my eye color. Okay, so the gene, essentially the gene is the genotype for my eye color. And let's say that my eye color, the color of my eye, when you look at me and you're looking at me and you're seeing that the color of my eye is black. So the color of my eye, if the color of my eye is black, then the phenotype for this gene is black. I hope you're feeling what I'm trying to say. See, gene is the genotype for my eye color. This particular gene is deciding my eye color, hence I call it the genotype for my eye color. But what my eye color is, what you can see my eye color is, what my eye color actually is, that is being expressed, how the gene is being expressed, that is the phenotype. So if my eye color is black, then the phenotype for my eye, for this gene, the phenotype of this gene is black. I hope you are understanding what I'm trying to say. So phenotype is essentially just the expression. When the gene is expressed, how is it expressed? That is the phenotype. And the genotype is the gene itself, like the genetic material itself. You can say that is the genotype. Now if you're taking another example, if this is the gene for my height, then this gene segment is the genotype for my height. And if my height is, let's say, 6 feet, then my phenotype is 6 feet. Okay, that is the meaning. Okay, I hope you're understanding. But we are going to go in depth into understanding what genotype and phenotype is in the chapter of principles of inheritance and variation, which is a very nice chapter. It took me a lot of time to understand that chapter properly because it's very beautiful. Okay. So the plasmid DNA confers certain unique phenotypic characters to such bacteria. So certain unique phenotypic characters to such bacteria, and one such character is resistance to antibiotics. So if there are two bacteria, one bacteria dies when we put a particular antibiotic in its medium, and the other bacteria does not die when you put the same antibiotic in the medium. And if both the bacteria are same, then that means that the latter bacteria, the bacteria that didn't die, was having a unique phenotypic character of antibiotic resistance to that particular antibiotic. Okay, so in higher classes you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. So this is plasmids are very essential in biotechnology chapter. It's studied in great depth in that chapter. Now, nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotes except for ribosomes. Okay, prokaryotes have something unique in the form of inclusions, and a specialized differentiated form of cell membrane called mesosome is a characteristic of prokaryotes, and they're essentially infoldings of the cell membrane into the infoldings. Means the cell membrane is the plasma membrane, as I said, right? Remember, cell membrane is the plasma membrane. So cell membrane is infolding into the cell, it's coming into the cell, and that is what is mesosome. Okay, now let's understand what is unique in prokaryotes and what is unique in eukaryotes. Unique means you can say this is a basic differences. Also you can call it differences between prokaryotes and eukaryotic cells. But what I'm trying to express is something unique. Unique in prokaryotic cells is something unique in eukaryotic cells. Oh shit, I sorry. Karyotic and prokaryotic. Okay, so prokaryotic cell, what do eukaryotic cells have? Most definitely they have unique, whatever is unique is their membrane bound nucleus. I'm sorry, membrane bound nucleus, and then membrane bound organelles. So membrane bound organelles are including many things like endoplasmic reticulum, Golgi apparatus, lysosome, vacuole, many things. It has a lot of things. Okay, membrane bound organelles. As we studied, organelles are essentially little organs because in a cell it is small, so little organs, that makes sense. Yeah. So in these membrane bound organelles, a lot of things are coming in, like endoplasmic reticulum is coming in, then Golgi apparatus is coming in, then lysosome is coming in, then vacuole is coming in, and then plastids plastids are coming in. So many things are coming in. So membrane bound organelles are like a lot of membrane bound organelles are there in eukaryotic cells, and it is only unique to eukaryotic cells, not present in prokaryotic cells. Okay, now let's understand what is unique in prokaryotic cells. Now unique in prokaryotic cells, first thing that we learned, you should try to fill this table yourself first, then check whether you have got all the points or not. Okay, so prokaryotic cells, what's unique in prokaryotic cells? First off, let's say plasmids are unique to prokaryotic cells. Yeah, plasmids are unique to prokaryotic cells. Then but one important thing is that not all prokaryotes have plasmids. Many prokaryotes have plasmids, but not all. Okay, but it is a very unique character to prokaryotic cells. Eukaryotic cells they completely don't have plasmids. Then inclusion bodies. Inclusion bodies, we are going to go in depth into what inclusion bodies are in the coming subtopics in the next page itself, right? So don't worry about it. Then inclusion bodies, and then mesosomes. So these are the things that are unique to prokaryotic cells. These are the things that are unique to eukaryotic cells. Okay, and mesosomes, to explain mesosomes, wait, let's just I was about to say that mesosomes are essentially replacement of membrane bound organelles. So whatever functions membrane bound organelles are doing, mesosomes do it for prokaryotic cells. Okay, because see, anything less than a living cell is not anything less than a cell is not living, and a cell can perform independent functions of life. Now in prokaryotic cell, they don't have any membrane bound organelles. Means these membrane bound organelles are things that are performing the essential functions of life for eukaryotic cells. But prokaryotic cells are also living, right? So even in them, something must be performing the essential functions of life. And majorly, mesosomes are the ones that are performing the essential functions of life in prokaryotic cells. Okay. So let's move on. Cell envelope and its modification. So okay, well, we have missed a diagram. Okay, so typical bacteria is 1 to 2 micrometers. PPLO, pleuropneumonia like organisms, are about 0.1 micrometer, and viruses are 0.02 to 0.2 micrometers, and typical eukaryotic cells are 10 to 20 micrometers. Now what are micrometer? What is micrometer? So 1 micrometer is 10 raised to minus 6 meters. 1 nanometer is 10 raised to minus 12 meters. Okay. Okay, yeah, one important thing I need to point out here is that notice how they are saying that a typical bacteria is 1 to 2 micrometers here, but in the same in the same chapter, just the previous page, you will see that they are saying that mycoplasma are the smallest cell having only 0.3 micrometers in length, while bacteria could be 3 to 5 micrometers in length. So 3 to 5 they are saying here, and here they are saying 1 to 2. So that is what I was trying to say, that it is completely, it's a range. It's not fixed, right? The length or the breadth or the measurements of these living organisms, it's not fixed. It's just a range. Even with PPLO, see here they are saying 0.1 micrometer, and here they were saying 0.3 micrometer. So it is a range. Okay, it is a range. It's not something that is completely fixed. Okay, now let's move on to cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound 3-layered structure with the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Okay, let's draw this. Okay, let's draw this. We already started drawing it. So innermost plasma membrane, which is the cell membrane. See, cell membrane and plasma membrane are the same things. Okay, cell membrane and plasma membrane mean the same thing. Cell membrane and plasma membrane are the same things. Now, innermost cell membrane or the plasma membrane, then the cell wall, and the outermost glycocalyx layer. Okay, glycocalyx layer, outermost glycocalyx layer. So that is the cell envelope. Together means the cell membrane plus the cell wall plus the glycocalyx layer. Together they are called the cell envelope for the prokaryotic cell, for that particular prokaryotic cell. Okay. Although each layer of the envelope performs a distinct function, they together act as a single protective unit. So if we are asked the function of a single, like let's say glycocalyx layer, then that is a different function, and cell wall, then that is a different function. But if we are asked what is the function of the cell envelope of a prokaryotic cell, like the whole cell envelope, so together they are acting as a single protective unit. So you can say that this single protective unit is a function common to all three. It's all three are trying to protect the cell. Okay. So bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Sir Graham, which is those that take up the Gram stain are Gram positive and those that do not are the Gram negative bacteria. Now that is very simple. So basically they are saying that bacteria can be classified into two types, which is Gram positive and Gram negative, and the basis of classification is that if you put the Gram stain on the bacteria, if the bacterial cell envelope retains the stain, then it is a Gram positive bacteria. If the bacterial cell envelope does not retain the stain, then it is a Gram negative bacteria. Essentially what happens is that we first stain the bacteria, then we wash the stain so that the stain is not overpowering. Only the things that are retaining the stain are colored and the rest is uncolored, so that we can actually see something, right? So if when we are washing the Gram stain off, if the cell envelope of the bacteria retains the Gram stain, then it is a Gram positive bacteria. If it does not retain the Gram stain, it is a Gram negative bacteria. Simple classification, not important. Now glycocalyx differs in composition, glycocalyx being the outermost part of the cell envelope. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, and in others it may be a thick and tough called the capsule. So glycocalyx has been classified into two. In some bacteria it has not been classified. There are two types of glycocalyx, you can say. Two types of glycocalyx. If it is loose, then it is a slime layer. If it is thick and tough, it is a capsule. And the cell wall determines the shape of the cell and provides strong structural support to prevent the bacterium from bursting or collapsing. Now this definition of cell wall is very important because it is true for all the cells having cell walls. And as I said earlier, most of the cells have cell wall. Like let's say plant cells have cell walls, fungi have cell wall, bacteria have cell wall. So only a few categories, a few types of organism don't have cell wall. The first common is animal cells. Animal cells don't have cell wall, and the second is mycoplasma. Till now we know that animal cells and mycoplasma don't have cell wall, but rest of the organisms have cell wall, and all the organisms having cell wall will have the cell wall will have the same function, that it determines the shape of the cell and provides mechanical support or structural support to prevent the bacteria from bursting or collapsing. And now imagine, I'll just try to explain it. Now imagine if a layer is preventing the cell from collapsing or bursting, then that layer itself must be very thick and rigid, right? And that cell itself, it is very thick and rigid. If that layer is determining the shape of the cell, then that layer must itself be very thick. True? Now in mycoplasma, since this thick layer is not there, there is no particular shape to the cell, hence it is called pleomorphic in nature. Yeah, then hence it is pleomorphic in nature, right? Okay, so the plasma membrane is selectively permeable in nature. Plasma membrane is selectively permeable in nature and interacts with the outside world. Now, what is selectively permeable? You probably know what selectively permeable is. It essentially means that the plasma membrane doesn't allow all material to cross the cell membrane into the cell itself. It chooses and only selectively allows a few material to cross the cell membrane, and we are going to see in which way does it choose, like which material it allows to go into the cell, which material it doesn't allow for to go into the cell. And that is, we learned that in the eukaryotic cells, because the structurally the membrane is similar structurally to that of eukaryotes. So even in eukaryotes and prokaryotes, the cell membrane is structurally similar. Okay, so in eukaryotic cells we are going to learn, see, about cell membrane. We are going to go in depth into the structure of the cell membrane. It's a good topic. Okay, so wait till then. Okay, so a special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. So mesosomes are formed by the extension of plasma membrane into the cell. As I said, let's say that this is the cell, this is the cell membrane. Let's say this is the cell membrane. If the cell membrane is infolding into the cell, do you get it? That those are infoldings into the cell. Then these structures are called mesosomes. Okay. These extensions are in the form of vesicles, means round or spherical, and tubule, which are tube-like, and lamellae, which can be branched. Right. So these infoldings, they are just saying that these infoldings, the infoldings into the cell, let's say these are the infolding. So if the infolding are branched, like if there are branches coming out of the infolding, then those type of infoldings are called lamellae, right, branched. And if the infoldings are tube-like, then they are tubular. And if the infoldings are spherical, then they will be vesicles, right? I hope you're understanding what I'm going to explain. Okay, so they help in, these mesosomes help in cell wall formation, DNA replication, and distribution to the daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. So as I said, they are essentially the replacement of membrane bound organelles in prokaryotes. So since prokaryotes don't have membrane bound organelles, but they also need to perform essential functions of life, right, because they are also living. So hence mesosomes are present, which perform most of the essential functions of life. See, look at the functions of mesosomes, right? It's like literally all the functions of life: DNA replication, distribution, so reproduction, and respiration, so metabolism, secretion, and all these things are performed by mesosomes. In some prokaryotes, like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Now these chromatophores, which contain pigment, they help the cyanobacteria to perform photosynthesis. And these pigments that they're talking about here are very similar to the pigments present in higher plants, which they use to do photosynthesis. And also notice how they're saying that there are other membranous extensions into the cytoplasm. Now these membranous extensions, they seem to be mesosomes, right? Because mesosomes are also membranous extensions of the plasma membrane into the cell. Membranous extensions of plasma membrane into the cell. And here also they are saying that there are other membranous extensions into the cytoplasm. So maybe they are a specific type of mesosomes, but for now let's just assume that these are completely different structures that just contain pigment, and these are also formed by extensions into the cytoplasm. Okay, so bacterial cells may be motile or non-motile. Motility just means if the bacteria is moving. If it is moving, it's called motile. Non-moving, it is non-motile. So if motile, they have thin filamentous extensions from their cell wall called flagella. If they are motile, then they have thin filamentous extensions from their cell wall to the called flagella. Now, what is the important thing to notice is that if the plasma membrane is extending into the cell, then they are mesosome, or they can be chromatophores, right? Chromatophores are also a type of extension into the cell. But we don't know whether plasma membrane is extending into the cell to form chromatophores. We know explicitly that plasma membrane extending into the cytoplasm are called mesosomes. And if the cell wall, which is the middle layer, if the cell wall is extending outside, then they are called flagella. Means it can be flagella or other surface structures, right? Flagella is usually, it's just one of the surface structures. There are many surface structures. It can be cilia, flagella, pillae, fimbriae, all these things. It can be, they're all surface structures. Okay, so bacteria shows a range in the number and arrangement of flagella. They're just saying that in different bacteria there will be different number of flagella and different arrangement of where the flagella is. That's also okay. Bacterial flagellum is composed of three parts, which is the filament, hook, and the basal body. Filament is essentially the thin, this long part is the filament, the hook like part is the hook, and the part that is attached to the cell is the basal body. Okay, so the filament is the longest portion and it extends from the cell surface to the outside. Besides flagella, pillae and fimbriae are also surface structures of the bacteria but do not play a role in motility. Okay, as I said, pillae and fimbriae are also surface structures. They are not infoldings of the plasma membrane. They are essentially extensions outside. It's coming outside. Okay, but it is not specifically written which specific which specific part of the cell envelope is extending outside to form pillae and fimbriae. But we know that flagella, if it is forming, flagella is always coming from the cell wall. But for pillae and fimbriae, we don't know. Pillae and fimbriae are surface structures, that we know. Okay, so the pillae are elongated tubular structures made of special protein. They function is not written, but function, as far as I have researched, it is essentially, it helps in the cell, in the bacterial cell, to transfer DNA from its from one bacteria to another bacteria. So bacterial DNA or genetic material can be transferred through pillae, and that is the function of pillae. Genetic material is transferred from one bacteria to another. That is the basic, function of pillae, and it is made up of special protein. So fimbriae are small bristle-like structures, bristle-like fibers sprouting out of the cell, and in some bacteria they are known to help attach the bacteria to rocks in streams and also to host tissue. So the fimbriae help the bacteria to attach to host cells or some any other structure that it wants to be attached onto. Okay, now let's understand very deeply what's going on here. Okay, let's say this is the plasma membrane. If there are infoldings of the plasma membrane, if there is an infolding of the plasma membrane, it can form mesosome. Mesosomes can be vesicle, tubular, lamellae, and if the cell wall is extending outwards, it is forming flagella flagella, and it helps in motility. Then there are other surface structures which we don't particularly know where they come from, where they arise from, which specific part of the cell envelope it arises from, but we do know that they are surface structures, so they are present on the surface of the cell, we do know that. Okay, so they can be pili, which are long tubular structures made up of protein, and it can be fimbria. Fimbria are like small bristle-like extensions. Okay, so I hope this diagram helps you to understand what this is. A funny diagram, but okay, I'm not good at drawing. Okay, ribosomes and inclusion bodies. Let's move on. Ribosomes and inclusion bodies. So ribosomes are also called pellet particles because it was discovered by Sir George Palade, and we'll learn about it in the coming topics. Sir George Palade discovered ribosomes, hence ribosomes are also called pellet particles. Ribosomes are made up of subunits. It's like one organelle. But it is made up of two subunits, each of the ribosomes, and there are two types of ribosomes. Okay, ribosomes. I'll just explain it here so that you can. Ribosomes. Okay, so ribosomes, there can be two types of ribosomes. One is 80S ribosome, the other is 70S ribosomes. 80S and 70S ribosomes. 80S is only present in eukaryotes, 70S is present in both eukaryotes and prokaryotes. And 70S is further, it's like one organelle, but it is made up of two subunits, which is the 50S subunit and the 30S subunit. And the 80S ribosomes are made up of 60S subunit and the 40S subunit. So they don't add up. I know 60S plus 40S should be 100S, but that is not how it works because this is not scalar addition. It is completely different. Some completely different. It's a totally different thing. S here is the Swedenborg's unit, and this is essentially this whole quantity, this 50S 70S, it is essentially the sedimentation coefficient, and S is the unit of the sedimentation coefficient, which is the Swedenborg's unit. Swedenborg's unit. Okay, we are going to learn in detail about all these things, but whatever I explained in the coming topics. In eukaryotic cells also we'll have to learn about ribosomes, right? There we'll learn about it. Okay, so now I hope you understood what ribosomes are. Essentially they are, you can say, made up of two subunits, and together they form one ribosome, and the two subunits can have different they are different in different cells. Like in eukaryotics they are different, prokaryotic they are different. Okay, so in prokaryotes, ribosomes are associated with the plasma membrane of the cell. That's very important. In prokaryotes, they are associated. Ribosomes are associated with the plasma membrane of the cell, because in eukaryotes, the ribosomes are essentially present on the endoplasmic reticulum. They can be present on endoplasmic reticulum, or they can be present in other organelles also, like mitochondria. They are also present in mitochondria, but in organelles, if they are present, they are 70S ribosomes. On endoplasmic reticulum, if it is present, then they are 80S ribosomes. So that is a very important point to remember, where exactly the ribosomes are present in which cell. So in prokaryotic cells, they are present. Ribosomes are associated with the plasma membrane of the cell, and they are about 15 nanometers to 20 nanometers in size, and nanometers, as I said, is 10 raised to the power minus 9 meters, right? They are about 15 nanometers by 20 nanometers in size, and are made up of two subunits, which is the 50S and the 30S units, which are together present to form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosomes of a polysome translate mRNA into proteins. Now, they are giving a lot of information even before the chapter in which the information is explained is being taught, because the chapter in which the in-depth explanation is given of what mRNA is, what is this translate into mRNA into protein, what does that even mean, that is written in molecular basis of inheritance, which is a class 12th chapter. But you have me, I'll explain what this is, not in depth but very briefly. Okay, so whenever any genetic material is dividing, so genetic material must divide, right? If genetic material itself is not duplicating, then the genetic material will become zero, right? Because if one cell is dividing into two, then the genetic material will become half. If the genetic material is not dividing, then it becomes 1/4, then eventually it will just become zero. But that is not possible, right? We need exact genetic material of the parent to go into the daughter cell, so that the daughter cell also can perform essential functions of life. So genetic material divides. Point is, genetic material divides. So during division of genetic material, I'll explain what happens. Okay, let's say this is the DNA, let's say this is a DNA. Okay, let's say this is a DNA. Now if the cell wants to, DNA is just a set of instructions. Okay, DNA is just a set of instructions. It does not, in itself it's not useful, but when it is converted, see, it's like, it's more like if you're given a rule book on life, but in a language you don't know, so that rule book in itself is not useful, but the translated information is useful, right? That is what is happening here. So let's say that this segment of DNA is coding for a particular something. So what do we call it? A gene. So let's say this gene. So this gene, if it is taken, let's say this is the gene. Let's say this is the gene. How is it made into something that can be used by the cell? Okay, I'll explain that. So this gene first self-replicates. This gene first self-replicates to form a copy of the gene, and that copy is what is used to make protein. Okay, first it is self-replicating. First the gene DNA is self-replicating, so that copy of the gene is used. Then the DNA is undergoes transcription. It undergoes transcription to form an mRNA. Okay, it's a simpler form. You can say it is a simpler form of the DNA, and then this mRNA is translated. This mRNA is translated to protein by ribosomes. So this translation is occurring with the help of ribosomes. So ribosomes are the one that is translating mRNA to protein. Sorry. So I hope you understand why ribosomes are important, because they help in the process of translation into protein. If there are no ribosomes, then this mRNA, because it's a different language, it's a totally different language. How can you understand how much ever useful that book of life is, how much ever useful it is, but if you can't understand it, what is the point? So to understand that, translation is done by ribosomes, and that is protein, and then it translated to protein, and this protein can be used in the cell to form whatever is necessary in the cell. Okay, so also mRNA is like a long chain. mRNA is like a long chain of, what do you say, instructions. mRNA is a long chain of instructions. Many ribosomes may attach to the single chain at the same time. Okay, many ribosomes may attach to a single mRNA to translate the mRNA. Then this structure is called a polysome or a polyribosome. Okay, this structure, this ribosomes attached to an mRNA, it's called a polysome or a polyribosome. Okay, so the ribosomes. Okay, I hope you understood. I tried to explain it, even though this chapter comes in like class 12th mid. So inclusion bodies help reserve material in prokaryotic cells are stored in cytoplasm in the form of inclusion bodies. They are non-membrane bound. These are not bound by any membrane and lie free in the cytoplasm. So essentially you can think that if the cell itself currently doesn't need a substance, but it may need it in the future, then that substance, that material is just lying around in the cytoplasm, and we have just given it a name, that is inclusion body. Okay, it's very kind of funny. Yeah, so example of inclusion bodies can be phosphate granules and cyanophacian granules and glycogen granules. They are all examples of inclusion bodies. Gas vacuoles are found in blue, green, purple and green photosynthetic bacteria. Gas vacuoles can also be found. So essentially, if gas is trapped inside the cytoplasm, if gas is not essentially trapped, means it is trapped, but it is useful. It is used for, you can say, buoyancy of the cell. If the cell doesn't want to drown, can be used for buoyancy of the cell. Okay, so gas vacuoles are found in blue-green algae, purple green photosynthetic bacteria. These are also inclusion bodies. Okay, so anything that is storing any material and is non-membrane bound, they're inclusion bodies. So prokaryotic cells, we completely finished prokaryotic cells, because in general prokaryotic cells were very simple. Prokaryotic cells are non-membrane bound organelles. They have a complex but chemically complex cell envelope. That is true, because they have a 3-layered cell envelope. They can form mesosomes, they can form flagella, they can form pili, fimbriae, all these things it can form through its cell envelope. And also they have ribosomes and inclusion bodies, which we have studied about in great depth. I hope you understood everything that was explained in the episode. Thank you. Have a nice day.